When scientists around the world are sharing information on a species, it's very important that they're all on the same page and talking about the same species. All scientists around the world use the same system for naming species. This system, called binomial nomenclature, was developed in the 1700s by Carolus Linnaeus. Linnaeus was an early naturalist who described and named thousands of the species on Earth. He set the foundation for how we name species today. But he is also a controversial figure for his views on race and the belief that human races constituted different species, which of course we now realize is incorrect and rooted in racism. As a result, many organizations that have named things after Linnaeus have updated those names to new ones that don't celebrate these early colonial ideas. Scientists all over the world use the same classification system, so we're all on the same page when we talk about a species. In this system, species are given a Latin name that is made up of their genus and species names. Humans belong to the genus Homo, which is Latin for man, and the species sapiens, which means wise. So our Latin name is Homo sapiens. This system of identifying species by their genus and specific epithet, or species name, is called binomial nomenclature. Bi means two, so this is a two-name system. The way species names are written are also standardized. We always capitalize the genus and write the species name in lowercase. We also always put the full species name in italics. So again, to correctly name and identify a species, you use the Latin names for the genus and the specific epithet, or the species name. The genus is capitalized and the species name is lowercase, and the name is written in italics. Pause now for another participation question. Using what you just learned, which of these would be the correct way to write the scientific name for this butterfly? So how do we come up with these different groups and organize species? We try our best to group them by relatedness, or their phylogeny. Phylogeny is the evolutionary history of organisms. We usually present this with a phylogenetic tree. This tree can tell us not just which organisms are most related to one another, but how they evolved over time from common ancestors. If we look at this phylogenetic tree, you notice that there are places where it branches off into different groups of organisms. Each node, or branching point, on the tree represents the common ancestor of all the species branching off from it. So the node that the green arrow is pointing to is the common ancestor of all the animals in the tree. It represents some extinct species that all these species descended from. The branching points also indicate to us that a speciation event occurred. A speciation event is a point in evolutionary history in which a population splits into independent evolutionary lineages. So members of that common ancestor species got different enough from each other that they were no longer interbreeding or able to interbreed. Maybe the population became physically separated by a mountain range, or some individuals adapted to eat one plant and others adapted to eat a different plant. And over time, they split into two different species. So let's take a look at a classic example of speciation, the finches of the Galapagos, known as Darwin's finches, because they inspired Darwin to develop this theory of how evolution leads to speciation. Darwin hypothesized that as the finches got more abundant and were competing for food with one another, there was no longer enough food for all of them. Over time, some of the finches developed adaptations that allowed them to eat different foods. So some grew larger beaks that allowed them to eat larger, tougher seeds. Others developed smaller beaks that were better for catching insects. As they specialized for these different diets, they developed into several different species. So let's take another look at this phylogenetic tree and make sure we can understand what it's showing us. Notice we have time on the vertical axis here. So if we look at common ancestor A, that is the species furthest back in time. Species A is the common ancestor of all the modern species in this tree. Then at some point, something caused a speciation event. An ancestor A split into fish and ancestor B, which is the ancestor of birds, humans, rats, and mice. Then, later on in time, ancestor B split into birds and ancestor C, which is the ancestor of all the mammals on this tree. So you've probably noticed that how recently two species shared a common ancestor can tell us how closely related they are. Our most recent common ancestor with fish was a long time ago, all the way back at ancestor A. But we share a much more recent common ancestor with rats and mice, so we are more closely related to the rodents, which are also mammals, than we are to fish. Now use this participation question to test your ability to read this phylogenetic tree. Now give this one a try. Are mice more closely related to birds or to fish? Remember that you can't always tell by looking at two species how closely related they are. Here's another participation question for you. Use the phylogenetic tree to determine which two animals are most closely related. So how do we figure out which species are related to one another and what their evolutionary history is? In early scientific history, we couldn't use genetics to tell how closely related species were. Instead, we had to rely primarily on their shape or morphology. But since the 1980s, we've started to classify species by their genetic similarity. This can give us more information about how related two species are. Look at the DNA sequences for the 4 species on the right. Notice how species 1 and species 2 have very similar DNA sequences because they share a recent common ancestor. But species 1 and species 4 have more differences because they split from their common ancestor much longer ago. Now see if you can interpret some DNA sequences on your own. Here's another participation question for you. Pause the video to figure out if species A is more closely related to species B or species C based on their DNA sequences. Notice that the gray arrows point out differences in base pairs between the sequences.